Hello, everyone. Nice to meet you all, see you all. I'm excited, as Rachel said, to share the big story of the little life and why we owe our existence to phytoplankton. So I've been at Scripps now, as Rachel said, since 2016. And in that time, I have become a polar guide, a maritime professional. I go down on tour vessels to run the citizen science program, which I'm going to tell you about. But I also do all the research in the lab and then, of course, use that data to contribute to my PhD thesis work. And so I've had this unique experience of kind of entering oceanography as the scientist side of it, but also through the tourism industry side of it now as well, and what that dynamic is like interacting with people who want to go see these incredible places in the world, and also how to talk about phytoplankton. So today I want to share with you how I fell in love with phytoplankton, and I want to share with you, of course, why phytoplankton are so amazing, and then also talk to you more about the inspiration and motivation for why I co-created the citizen science project. So before I get into it, who knows what phytoplankton are? 2, 3 people. 4. Who has like recreates or works on the ocean or with the sea? Okay, got a lot of landlubbers here, and so hopefully by the end of this, then you might have a favorite phytoplankton you can go home knowing about. So how did I first get involved in polar and Antarctica? It seems a bit niche, right, in phytoplankton. So I, in high school, actually thought that I liked traveling, and the humans that travel the most are astronauts. So if I became an astronaut, then I would go to the moon and I would travel everywhere. And in doing your research for those careers, you realize astronauts don't actually spend all their time traveling to the moon. It's mostly on land and training. But I saw that astronauts get their degrees in science and technology and math. And so when I was thinking about my life beyond high school, I went to the University of Washington and decided to major in STEM. I chose biology and geology, but mostly I wanted to go to University of Washington because I was from Washington State, so it's in-state tuition, and I really wanted to be in the marching band. So I play clarinet, and I thought they were so cool when they ran out on the football field. So, and I had a friend going because no one in my family's gone past high school. So for me, I was like, do I need to go to college? I don't know. But I really wanted to be a Husky band member. So I also this time, you know, there's this saying like jack of all trades, master of none. And so I had a lot of anxiety and figuring out like, what do I want to be when I grow up? And I'm going to pick the wrong degree and then seal my fate. And I won't get to do what I think I might want to do in life, but I don't know what I want to do in life. And so it was, I wish I could talk to my younger self now, because what I ended up doing was choosing biology and geology because I didn't want to choose anything specific. And in doing so, picked a bunch of different jobs where I worked in immunology in the lab and taking care of mice, and realizing I don't really like those questions scientifically. I then was a field biologist. I went and chased squirrels in the Cascades of Washington, looking at how forest fires influence where they make habitat. I went to Peru and Mexico to study parrots and macaws and conservation efforts. I also went to Africa to lead trips with undergrad students to study big cats and wild dogs, and how these big predator animals are influenced by the landscape and influence the landscape. And then finally, I settled on a job back in the lab, but this time with questions focused on phytoplankton and how does increasing carbon dioxide levels in the atmosphere affect these single-celled organisms, which at that time I didn't really know anything about. I knew about the cliffs of Dover. Have you heard of those in south of England? And I had heard of diatomaceous earth. Maybe you use that in like an insecticide to kill insects. And so I was like, yeah, I could pick up something about these phytoplankton things. And ended up working at this job in Seattle for 6.5 years as a technician, running, basically torturing phytoplankton with lots of carbon dioxide and seeing how they responded at a genetic level. So to me, that was really cool to be able to be in the genetics world, but also stay in the ecology world, but have a environmentally large scale question that we were asking. And also I got to still go into the field. And the biggest opportunity I had during that job was in 2013. My boss at the time said to me, she was about to let me go because she didn't have any funding. And instead, a month later, she came and said she got funding from the National Science Foundation with a team of scientists to go to Antarctica. And she didn't have the time to go, so I was going in her place. I knew nothing about oceanography. I had always thought maybe someday I'd go to Antarctica as a traveler, a tourist. I had heard that that was growing. And I was to spend 53 days on this icebreaker. It's one of the United States has two. They're soon going to only have one. The Nathaniel B. Palmer, and we were going to study this green stuff that grows in the sea ice. And the project was called Tracers. We were going to trace the fate of algal carbon in the Ross Sea. So how do algae or phytoplankton grow, and how does it sink down to the seafloor and then come back up in through the microbial loop and then get into the atmosphere, and kind of put that whole system together with the research team. And again, I knew nothing. I'd never been to sea. So I, in fact, in college had done a geology field trip where we dredged something up in the San Juan Islands, and I thought to myself, this is miserable, it's wet, it's cold, it's windy, I will never work with the ocean. That's what I told myself back then. So here I am on a C-130 Hercules flying down from the US to New Zealand, then to Antarctica, and we went to one of the US stations called McMurdo. So there's the South Pole, McMurdo, and Palmer Station, and I was doing the work out of McMurdo. It's the largest base, and it has 1,000 scientists in the summer and maybe 100 in the winter, whereas the other US-based Palmer is 100 in the summer and 10 in the winter, so just to give some scale. And when you get off the plane, they're like, everybody put your extreme cold weather gear on, it's really cold. And I was like, yeah, cold. Like, what, Alaska cold? It was so cold that as soon as I pushed the trigger on the camera to take a picture, my finger was like feeling frostbitten. So I was like, okay, it's a desert. It's desert cold. There's not a lot of precipitation. You immediately feel like you need chapstick. You can feel the moisture leaving your skin. And then also, I had always thought Antarctica would be like flat. When you think of Antarctica as mostly like South Pole expeditions. And so when I looked around, I saw this amazing mountain range, and there's volcanoes and Mount Erebus. And then you think like it's part of the South American Andean mountain range. So I don't know why I had never considered that there would be mountains in Antarctica, but it was gorgeous. And I felt like an astronaut having just landed on another planet. But I was like, this is on Earth. And so I decided I'd dedicate my whole career to studying Antarctic ecosystems in some way. But in 2013, I still was never going to go to grad school. I didn't think I wanted to have that as part of my process there. So either way, I was hooked, and I didn't know how I was going to make another expedition happen. But this was in our 53 days. One day we were able to jam the icebreaker deep enough into the sea ice so that we could get out. And I don't know if you guys know Jerry Koyman at Scripps Institution of Oceanography. He's a penguin biologist. He tracks diving and physiology behavior of emperor penguins. So the one day we got to get off the ship was to go chase emperor penguins and put dive trackers on them, which was incredible. So this is what the Ross Sea area kind of looks like. It's the ocean moving. You can actually maybe see two little penguins floating around on it. This is when the ocean is salty, so it freezes at around negative 1.8 Celsius. And so we call this slush ice. So it gets kind of slushy looking. There's also other names like grease ice and pancake ice. And so, you're on a ship where the food is declining and all you're thinking of is like food related ideas to the ice. So pancake ice with maple syrup, and like, it gets pretty stark. But the idea was to try to understand, again, these phytoplankton. And so do you guys know this character? Okay. This is most of what people think of when they think of plankton is the SpongeBob cartoon. So this is named Plankton in the cartoon, but it's really a copepod, or what we call a zooplankton. So if you're to break up the words, they originate in Greek. And so phyto means plant, and plankton means drifter or wanderer. So a zooplankton is an animal, animal plankton, animal drifter. So it's anything that can't swim away from the current. So technically jellies or mola mola fish are plankton. They'd be animal plankton. And so when people think of phytoplankton, they think of that thing, but it's really, anyways, it doesn't translate well. I think uh, phytoplankton are more like this. So you see two different communities of different collection of phytoplankton species, and they also come in these different size ranges. So this is some phytoplankton, it's a generic term. It's basically like any single cell that floats around in the ocean that can photosynthesize and use sunlight to make energy is a phytoplankton. And you can have phytoplankton that use cellulose wood to make their cell structure. You can have them like this one uses silica glass. We call them diatoms, glass houses. We have flagellates, or little phytoplankton that have flagella and can actually swim around and move. And then you have, this is another type of diatom. It can actually slide against each other, and so it will slide along surfaces and kind of move. And you also have plankton like the cliffs of Dover that are made of calcium. So trying to understand all these different phytoplankton types, I mean, to us, it's like, oh, they're all the same. But they're actually very complex and have their unique behaviors, so to speak. So I'm going to admit to you guys that I always used to think the ocean was boring. That's why I was like, boring, cold, not going to do it. Like, nothing's going on there. And when I was on the ship in Antarctica, the chief scientist was giving oceanography lectures to his students. So I sat in on them, and that's where I learned that, Antarctica, when the ice freezes, it's generating this really salty, dense, cold water that sinks to the bottom of the seafloor. At the equator, we have the sun that's evaporating because it's so hot, and that sets off this huge conveyor belt. And so the ocean is actually layered and structured, and you have phytoplankton that like to live in different layers. And then you also think about, like, to us, water is very fluid. We swim through it really easily, but to a phytoplankton, it's viscous. It's like they have to figure out ways to move through this thick solution from their single cell perspective. So a lot of times people ask me, like, what do phytoplankton eat? It's similar with plants, right? You guys probably already know this, because plants, they're floating solar panels. So they eat light, they eat photons, they eat carbon dioxide. They can get that from the water passively, or they can pump it into their cell. They eat basic nutrients just like plants. So nitrates, phosphates, silica, iron, trace metals, vitamins. They hang out with bacteria to get the vitamin Bs. Like, there's like similarities. And who was the first phytoplankton to figure out how to photosynthesize? And have you guys heard of cyanobacteria? Or it's actually a superfood now. You can eat cyanobacteria. It has omega-3. It's very healthy. Or blue-green algae is another word for it. So cyanobacteria, the earliest known fossils are from the Archean era, 3.5 billion years ago. So whatever this single cell organism was doing in the ocean, it figured out how to split water to make energy. So use carbon dioxide, light, and the byproduct, it doesn't need oxygen. So it's just putting oxygen back out into the ocean. I like to think of things chronologically. So this next figure, we're going to walk through it together. I found two timelines and tried to stack them on top of each other. Actually, does this have a, yeah. Oh, okay. It works on the wall. So at the top we have atmospheric oxygen. In present day, we know we're around 21% oxygen. And at the bottom, we have the oxygen being accumulated, showing the bluer it gets, the more oxygen there is. So the higher this blue line goes, the more oxygen there is. And if we start at the left side, in billions of years, we can see different markers. So those stromatolites I talked about, the microbial fossils around 3.5 billion years ago. Then cyanobacteria for sure was found on the scene. And that gave rise to the appearance of oxygen producing photosynthesis. That was accumulating in the ocean. In the geological record, there's something called the Great Oxidation Event. And then we get the diversification of cyanobacteria, more fossils being found. And then this one jumps to dinosaurs. I think we all like relate to dinosaurs. So around that same time, you see a big spike. And when we think about what's happening, we zoom in on that red section. We're still looking at the atmospheric oxygen. we had in the ocean, this is now millions of years, so 600 million years, which on that time scale is relatively recently. The Cambrian explosion was in the ocean. That's also a big fossil record marker of all these marine animals. But then you see land plants. The first land plants started to come onto land around 420, 450 million years ago. So a lot of that oxygen production is set up because of the phytoplankton in the ocean, or these cyanobacteria. And if we think about light on land and what plants are looking at, they're looking at visible light spectrum, so they can kind of access that whole visible light spectrum. But do any of you dive or go underwater snorkeling? Do you notice the color disappears? What color disappears?
Red.
Red kind of looks black almost underwater, right? Yeah. So water absorbs photons. It absorbs light. And the first, if we look at this wavelength spectrum, as we're going from the surface to depth, the first kind of wavelengths that are being absorbed are the reds. So you kind of don't want to choose your dive gear in red color scheme. But that's also why the ocean looks blue back to us when we look at it, because blue is not being absorbed by the water molecules in the ocean. And then if you're a phytoplankton, you got to think about, you want those photons and the water is taking it away. And so how are you going to figure out where in the sunlight spectrum you're going to get access to photosynthesize? So, one of the techniques that single cell organisms use is called endosymbiosis. So have you heard of that? Endosymbiosis theory? So this is where you had your two independent bacteria. So we have our cyanobacteria, and then we have another bacteria. And it's a crazy world in the single cell world, especially in the ocean. So one bacteria just engulfs another, like maybe you've seen videos of amoebas like scooping up things. And now they're like, oh, actually, I don't know if they're I'm anthropomorphizing. Like, actually, we want to live together. So they hang out. That's also how you get eukaryotes, right? So you have prokaryotes where just the single, and then the eukaryote cell has the organelle from another organism now as one. They realize it benefits the both of them to be together. And because the way they reproduce is by asexually dividing and just, exponentially growing, that gets passed on generation to generation. In the phytoplankton world, so we go back billions of years ago when we had our cyanobacteria, it got engulfed by another bacteria hanging out in the ocean. And that gave rise to three main types. So we had glaucophytes, which you don't need to worry about. We have red algae and green algae. The green algae and the red algae continued to get engulfed secondarily, so secondary endosymbiosis theory. And then the green algae is what gave rise to land plants. So all land plants are descended from green algae that came from the ocean from a cyanobacteria being engulfed by another bacteria. And the red algae continued to get engulfed. And the way, like, this team is really good at talking about this, Keeling 2004. So that was a lot of work done because of genetics. So we were able to see these are like Russian dolls stacked in to each other because of the genetic differences. So as scientists, they're looking at like, oh, well, these ones are green and these ones are whatever color, and this one is made of chalk and that one's made of glass, and or this one eats this, or that one specializes in this. And then when we brought in the genetics, it's like, oh, actually genetically they're finding, these different relationships that they didn't know existed before. So the story got a whole lot more complicated. When we look at the tree of life, they sit here, phytoplankton, they're like protists. They're not really bacteria, they're not archaea, they're not really plants, even though phyto means plant. They're not fungi, there are fungi in the ocean, and they're not animals. So there's like a weird collection of what I think are very sci-fi organisms living on this planet. And that's also probably why like NASA's big into like space and search for life, and a lot of genetics and phytoplankton single cell work, microbes might help us discover that on other planets, or relate some of our knowledge in that way. So back to the light. We have all these crazy single cell phytoplankton trying to compete with each other. And they also want to compete for this light. And so they'll build these accessory pigments around that can capture these different wavelengths of light. And then they kind of shuttle it to the photosystem, where photosynthesis is happening. And that's how they can choose kind of their niche to be efficient in. And we can measure those pigments and look at the light absorption spectra for different marine algae. And you can see that chlorophyll A is on there. So every plant or algae would have that chlorophyll A. But you also get chlorophyll C, B, you get carotenoids, you get phycocyanin, phycoerythrin. All of these accessory pigments are trying to capture photons from the other spectra so that they can be competitive in the ocean. And when we put all the like spectra together and see this certain collection of different pigments, you can actually relate a tree. So if you look at the background color here, you see that on the far left, cyanobacteria, they all kind of, all these different pigments. If you found those on your instrument, you could say you have cyanobacteria in the sample. If you had the green background ones, prasinophytes and chlorophytes, that's green algae descended, that would be a certain collection of pigments. And if you found some that were more related to the red algae lineage, or dinoflagellates, chrysophytes, diatoms, cryptophytes, prometiophytes, like they're all, you could kind of say, you know what's in your sample based on a pigment analysis. And I feel bad for phytoplankton because they kept their Latin and Greek names. But plants get tulips, daffodils, cedars, you know, like nobody's renamed phytoplankton, but one dinoflagellate means whirling whip. What if we were just like whirling whips? Corythron means broom, like broomstick. So it's like diatom means cut in two. So yeah, I feel bad because nobody, I feel like they're not relatable because they still have, you could say, I speak Greek if you learn some of these phytoplankton names. So, so we went over what they eat. They also, if they're not good at photosynthesizing, they can eat each other, like we talked about with endosymbiosis. So my favorite phytoplankton is called the dinoflagellate group. It's the whirling whip, and they can be selectively feverous. So if they find a chain of diatoms that are nice and omega-3 lipid rich, then they can cast a mucus net around them and then dissolve them and slurp them up. They can also stick what looks like a straw, it's called a peduncle. They stick that into the cell and slurp out the insides, and they can steal the solar panels of another phytoplankton and then photosynthesize themselves. So that's called kleptoplasty. It's like weird. There's also a dinoflagellate where they found the genome was 100 times larger than the human genome. And you have to wonder, like, what are these things set up to do? Like, why do they need that much genetic information? So we're going to move that over there. I had mentioned they asexually reproduce, and they also sexually reproduce. So if we just look at two examples of phytoplankton, there's the diatoms and the dinoflagellates. And they start off as this adult vegetative state in the ocean, but then they'll divide and divide until they get to a certain size where they can't physically like divide anymore without getting too small. So they kind of balloon up into this big oxospore, and they release gametes. So then somehow in the ocean, the gametes find each other. That's the sexual reproduction stage. And then they'll, in dinoflagellate case, they go into a resting cyst or a spore, and then they'll eventually, if nutrients come back or conditions are favorable, they'll go back to the vegetative state. So phytoplankton, when they're doing this and they're growing in whatever stage they're in that life cycle, we call it a bloom. So just the same way that wildflowers bloom on land, when phytoplankton are growing, it's a phytoplankton bloom. So have you guys seen the bioluminescent bloom that happens on the coast? So that's a particular type of phytoplankton that just has a reddish pigment in it. In our coast, that red tides are what are also bioluminescing. In other parts of the world, that's a toxic species, but here it's not. So it's, I mean, you see people swimming in it still, and there's other problems that come with large blooms, but it changes the color of the water, and you can see that color change from photographs. So if you were to look, that's the pigment differences. So depending on which phytoplankton are blooming, different pigments change the color of the water. And you can also see that from space. So satellites are going around space kind of detecting this chlorophyll, trying to see where you see purple and blue, there's not data or there's no growth. Where you see light blues, greens, The orange and red are where phytoplankton are blooming. So this is a picture of the Antarctic Peninsula. You have South America, just the tip and the top of the image. And then the Drake Passage, which if you go down on a tour ship, has anybody been to Antarctica? You have? Yeah. Did you cross the Drake?
Yes.
Drake Shake or Drake Lake? Okay. So when you get to the peninsula, then were you looking for phytoplankton? No. You're going for like whales and penguins, the boring stuff. Yeah. So this is an example of how we can use remote sensing and chlorophyll to kind of understand where phytoplankton are growing. But also in this image, you see there's a big patch of like gray. So in the polar regions, they're just always covered in clouds. So it's really hard to use remote sensing alone to understand those parts of the world. So this is a composite of chlorophyll measured in the northern and southern hemisphere on land and in the ocean. And right now I want us all to take a deep breath. Okay, that one was from your plant friends, our plant friends on land. Let's take another breath. That one came from phytoplankton in the ocean. And we say that over half of Earth's oxygen is produced by phytoplankton in the ocean. And if you want to get into the specifics of what the actual quantity is, it depends where you are in the world. But you see in the northern hemisphere, a lot of the vegetation and a lot of the land is up there. But in the southern hemisphere, when winter comes in the north, it's summer in the south, you see a lot of the chlorophyll or oxygen production would be in the Southern Ocean. And then you can also see where you get a lot of phytoplankton in the ocean. That's where a lot of the world's fisheries are. So this is forming the base of the marine food web. This is food for the animals in the ocean. It not only oxygen production, but I love this because it's like the two lungs of Earth breathing. So, so back to our phytoplankton basis. The other thing that they have to watch out for is they want to avoid death. Like nobody seems to like to die. So phytoplankton don't either. And in the ocean, it's crazy. They're viruses. They'll grow and eat all the nutrients around them. So they try to find ways to like collect nutrients around them or move through it to new patches of nutrients. The viruses will try to attack them. So what kind of defenses could they have against a virus getting in to explode their cells, basically? There's something called programmed cell death, which a lot of scientists try to understand is like, why would a cell basically cause itself to die? What's the benefit of that? So there's a whole line of research there. And then also grazing. They don't want to get eaten by those zooplankton that I talked about. So a lot of them, like cactus, a lot of them will build these spines and try to keep this hard, what we call a frustral as their cell wall. And they don't want to also get too heavy because then they'll sink out of the sunlight layer. They don't want to leave the sunlight, but that also is a way to bring phytoplankton or this nutrients carbon biomass to the seafloor if they do sink out of the sunlight layer. And then advecting is like also if they move out of a patch, they can't swim out of the currents, so they'll just get moved around. So we go back to Antarctica, and now you might be wondering, like, there's no trees or plants there. So how do we think about this down in a region that seems so desolate? And one of the really cool things that happens in Antarctica every winter, the sunlight disappears for 6 months. So in that time, it gets the ocean cold enough, that negative 1.8 Celsius, to freeze the surface of the ocean. And that sea ice that's kind of around the continent doubles the continent in size by area. And you can see where I was McMurdo Station, and this is the Ross Sea, and it's very sea ice filled, which is why we were there trying to study that. On the peninsula, so the part where you would come from South America and cross the Drake Passage, that peninsula sticking out, not so much sea ice, so kind of a different ecosystem down there. But what happens with this sea ice, as soon as the sunlight returns, you get this bloom of phytoplankton happening under the ice. So if you had the microscope, this is actual, not my hand, but actual microscope photo of the phytoplankton in Antarctica. And that's really important because were you guys who went to Antarctica, were you surprised by how much wildlife you saw? Or were you like, You could see more. Was it a good surprise? So it's, yeah, maritime. It's a maritime food web. So you have this is a short food web as well. So it's sunlight to phytoplankton. Phytoplankton, we talk about a lot, the krill, Antarctic krill eating the phytoplankton, but there's also fish, and any larval stage of a marine animal is eating phytoplankton. And that krill is the key food source for the seals, the penguins that live down there, these migrating whales that come from the equator, Arctic terns, which come all the way from the Arctic and fly down to Antarctica just to eat krill. And so it's really important to try to understand this link between not just krill and is it there or not there, or declining or not declining, and there's also a fishery for krill, but what about the krill's food? What's happening to the phytoplankton? So krill are very picky eaters. And in this cartoon, there's kind of demonstrating, one krill says, "Oh, diatoms, they're my favorite." And the other krill has a bouquet of diatoms saying, "I know, they're in bloom." So this is Jen Walsh is actually at NOAA, Southwest Fisheries Science Center. She does these cartoons online if you want to follow her. But I just love them because they're silly, but they also have truth to them. So a krill has these little, in the cartoon you can't really see it, but these little comb-like feet in the front. Have you guys seen like a crab feeding? You know, the crab's like blah, blah, blah. So it's kind of what krill are doing. They're combing phytoplankton out of the water. And they're looking for large, centric, circular, or chain-forming, lipid-rich, omega-3 rich diatoms. And so if you think about how the phytoplankton, if that's the base of the food web, might be changing, and you find smaller phytoplankton, is that enough for these krill to survive on? Are their young krill, juvenile krill, going to be able to feast on enough of that phytoplankton or diatoms, sea ice algae growing under the ice when they're in their younger years? And this image is trying to look at, if you make a color a certain phytoplankton, kind of like in that pigment thing I showed you, and you just count how many are in a parcel of water, and relative to what you counted in that parcel, 0 to 100% of things you could have counted from the season, which is opposite of ours. So their summer is November through March. What do you count the most of? You'd say, oh, well, actually the blue are small flagellates, those tiny ones. So by count, we're seeing a lot of these small ones. But every now and then you get a punctuation of red, which are dinoflagellates, or yellow, diatoms, or this light green, prasinophytes. So you're wondering, like, when would that be happening? Who's there, and why might it matter? Because by count, you might think these small flagellates are important, but there's ways we can estimate based on the size and shape how much carbon the phytoplankton are contributing. So when you estimate the carbon, you see that most of it is coming from the gold, the diatoms. So diatoms are contributing the most amount of carbon to these ecosystems. And every now and again, like where you see this red line, it shows up. So in that sense, the high number of smaller flagellates or dinoflagellates is also contributing carbon. But kind of piecing this whole story together is our mission right now. And this is taking you guys down to the Antarctic Peninsula. So that region where there wasn't a lot of sea ice, kind of different from that first expedition I had done in the Ross Sea. This is fjords coming out of the water, amazing mountains, glaciers that just come straight to the ocean. You don't really see that in the Arctic or elsewhere in the world. So it's just an incredibly massive scale. And I think maybe people have a hard time, like, you feel small when you're there. And I think it's hard to like really be in the environment. So, but it's amazing. It's like, again, it just hooks you. And so you get kind of this addicted to this ecosystem. So why would we want to study the Antarctic Peninsula? Why do I want to study it? We know that it has some of the fastest rates of warming. So not just from increasing ocean temperatures, but also air temperatures. So if you're a glacier and now your ocean is warming and your air is warming, that's a double effect. So 87% of these glaciers are retreating from the peninsula. This is a figure that was actually from a National Geographic article that shows where the purple is expected retreat, where the bedrock will be exposed. So what's that going to look like when you now have bedrock and new habitat that wasn't ever there before. What is that going to do to the penguins that build their colonies and all these yellow dots? Up here it's kind of small, but the krill is in red, so there's a big like river of krill. The krill hang out and they go feast all in this area here. It's also where a lot of the fishing ships are going, and the fisheries are out in the more open ocean too. And like I had said, we're trying to use remote sensing capabilities, which are needed, and that's a good composite image showing chlorophyll. So the phytoplankton where they're blooming, when you get enough days together, you get the clouds don't really influence the signal. But one thing I learned is the tourism industry is growing. So scientists from all the nations, there's maybe 10,000 researchers. It might be more now, maybe up to 20,000. It was hard to get an exact number, but 10,000 at least from all the nations of polar scientists going down to Antarctica. In tourism, this is an older publication, but currently the last season was over 100,000 visitors. So that's more than 10 to 1 people visiting. And ship growth. So after the pandemic, there are 21 new ships built to go explore because it's a place people want to see. It's amazing. And so ship traffic is also increasing. So that was, as somebody who'd been to Antarctica, wants to dedicate my life or my career to studying Antarctic ecosystems, and also really loves outreach and getting people involved and excited about not only science, but also the places they're visiting. I was like, why don't we build a citizen science project? So that's how I had met Rachel, and we had spent a 1 year just like, what do we want to do with our 1 year here? And I worked together with Maria Vernay at Scripps Institution of Oceanography and researchers in Argentina, the Universidad La Plata, to create a citizen science program or participatory science program where we train the guides or the expedition staff that work on the ships how to do a various collection of sampling with travelers. So you could be on a tour vessel and participate in this 1 hour activity where we get these samples. And I'm going to play this for you.
Waters, and a team of scientists are determined to uncover the mysteries that lie within.
But to understand what makes this ecosystem come alive,
FjordPhyto is a citizen science project collecting phytoplankton.
phytoplankton are microscopic algae that form the foundation of most marine. And by looking at how phytoplankton communities, researchers are studying how it might relate to other creatures. Because to better protect an ecosystem, we need to better understand it by protecting the microscopic heroes of the Antarctic and life that depends.
Because conservation is not a dog. It's a weed.
So that's a glimpse into FjordPhyto. We're looking at phytoplankton in the fjord systems of the Antarctic Peninsula. And of course, this isn't done by one person. So we have teams, we have students, we have a film student that made that little 1-minute trailer. We have satellite people, we have artists, we have social scientists and educators. So our team is growing and is one of the reasons this collaborative effort has been possible. So sometimes myself and Martina lately, Martina's my colleague here from Argentina, she and I have now become polar guides that can also lecture on the ships and get hired to work as staff if we want. It's hard to also do your research when you're being a polar guide, but we make it work because we'll train staff and when we're on board, and we'll also engage in doing the project. So what you would do is we provide a kit of gear, and that would include a phytoplankton net. So you'd tow that behind the boat. Secchi disc is just a disc. That's a quick way to determine the light level when it disappears or how much growth there might be happening in the water. If it disappears quickly, there's a lot of growth. If it disappears, you can see it for sometimes 20 meters. I saw it, then it's not much growth happening. We give them this instrument here we call the CTD. It stands for conductivity, temperature, and depth. So that device can go to 100 meters, and it has a chlorophyll sensor on the side of it. And it's measuring how salty the water is, what temperature it is, where these phytoplankton chlorophyll signal might be the highest. And then we have physical bottles we give them to take samples that will then count under the microscope, process for DNA in the lab, and also get analyzed for oxygen isotopes. Did the fresh water we see come from glaciers or sea ice or just water? And over the years since 2016 now, we have had thousands of people participating. So I love getting, when I'm in the thick of like writing, I'll get WhatsApp messages from the guides working of them having selfies or people doing the project, and it's really fun to see that and also get a chance to go down. And what's happening is, chlorophyll can tell you a lot or a little, and even when you collect it from the water, you can see a lot or a little. But if you look at these two dark chlorophyll samples, again, that community is totally different. So that matters for the krill. So that's why we're trying to put all these pieces together, pieces of the puzzle. If you were to look at that CTD that we dropped from 0 to, in this case, 80 meters, this chlorophyll is around 10 micrograms per liter. That one is the lighter water that you saw, 0.2, but the signal's the highest, just under 5 meters. So it's kind of like what's happening in the water that might make it so that you get these different profiles. And how are we going to compare that with what we see in the microscope and also with satellite? And that also, getting the physical sample can help us understand like how are the phytoplankton behaving, because in this case we saw this little horned looking basically a spore or cyst formation. So it's getting ready for winter. It's going to go into kind of like hibernation mode. And that's really cool to see, because some of these phytoplankton, there was a paper that came out that said it's really hard to tell a cyst and which phytoplankton it actually is. There's a paper that said diatom resting spores can survive in nature for several millennia. They had a spore that was out of sediment, and it revived after it was dated to be 7,500 years old. So it's like, okay, if things are changing and these spores are on the bottom of the ocean, like how might that change things in the future? So things to think about. This is an example of under the microscope, but then I get all these back in the lab. So I don't actually get to see them in a pretty form. I just see them in like a slushy form. And over the 6 years, we've sampled 39 different locations along the peninsula where all the tour ships are visiting. They go repeatedly over the 5 months of the season, getting thousands of people involved. And then we present all this as much as we can out in the public and at conferences and papers. And this is back in the lab. I pull out those filters that they towed the net and filtered the phytoplankton. And Christian and I, he's the newest PhD student. I warned him work-life balance is going to disappear, but he doesn't believe me. And so we process these samples. And then our colleague Martina has come on fellowships to work with us at the James Craig Venter Institute, which is near UC San Diego campus. And we extract the DNA, we extract the genetic material, we try to look at identifying species from DNA, but also their genetic behavior or response to the environment they might have been in. Martina takes the samples in Argentina with her team and counts them under the microscope. It takes her 3 to 4 hours to do each sample, and she has hundreds. So she's done with her PhD. She became a doctor, so she used a lot of that data for her work. And she takes amazing high-powered electron microscope images. And she's been able to see some phytoplankton that had never been seen or documented before. And this potentially could be a new species, but there's a lot more work that has to go into validating that it is a new species. But because the ships are there for so long, we can capture these finer scale things. And then also we can put together, I was saying earlier, I think about plant succession a lot, because in the plant world, it's like, okay, a fire comes through and takes out the plants, or there's these disturbances that happen. And so which plant species come back to populate that? But I'm thinking of it now in the ocean context. Like you have a 3D watery world environment. What's a disturbance? What succession do you get? Like there's a baseline community of phytoplankton. That's the basic assemblage we see. And depending, in this case, it was a warmer water and a colder water area, north and south. We had a different kind of blooms growing. So the beginning of the season starts off with this corythron, small centrics, but then in that case you get small flagellates blooming first, and here you got larger diatoms blooming first, and then who transitions is totally different. So with all that data from the peninsula, that's what I'm currently putting together for my PhD thesis, which I have to finish in the next 20 weeks. So I just got the letter from school. So we also work with a satellite postdoc doing satellite remote sensing. So all the physical samples with the oxygen isotope are being used, and the chlorophyll measurements, to validate this algorithm he's building that can use remote sensing satellite to detect where melting glacial water is coming into the peninsula. So he's been having work published on that. Now, all of this isn't going to be as useful unless we make it fair. So fair is findable, accessible, interoperable, and reusable. It's a new push to really make science accessible to other scientists, but also the public. So we're working with, it's a lot of work to like curate your data sets and make them actually usable. So we've published our first year of data, and we're working on cleaning up everything else so that somebody could just go to this page and see everything that's associated with this project. And then we do a lot of ocean literacy and education. So we have iNaturalist. This has kind of been still in pilot phase. If anybody uses iNaturalist, I know for plant identification, you can also use it for phytoplankton. You just have to make sure you get one phytoplankton in the picture, which is hard. We're developing an identification guide. So guides and travelers on the ships, if they have microscopes, a lot of the ships do, they can identify. We've built this book like you would a plant guide or a tree guide, or we're doing the same with phytoplankton that we find in these samples. And then we're able to, through some funding, take this work to Puerto Rico. So last summer and this coming summer, we're integrating with a NASA team called Oceanos that's raising maritime topics with students in the tropics and Spanish-speaking communities. So they're like, why is Antarctica related to Puerto Rico? That's exactly why we're going to tell them how it's related, and these migration patterns and phytoplankton and the ocean and how it's all connected. So just looking ahead, we're completing this field season. It's wrapping up. All the ships are leaving Antarctica now. We'll data, analyze that, try to make it fair. Not try, but we will make it fair. It's just a process. I'll finish my PhD. We'll design a new training platform to keep expanding and get really rigorous training of the guides that work down there. That's a huge component. And establish the program to be sustainable and continue growing. Find ways to do that. So I hope that's a whirlwind, but that you see that this invisible forest is tiny but mighty, that phytoplankton are so incredibly important for carbon cycles, for oxygen production, for the marine food web. They are crazy sci-fi stories if you want to dig into any rabbit holes with that. And I hope maybe you might have a phytoplankton now that is your favorite, and you can go home and remember to tell people about it. So thank you.